Hello. We are now going to do Chapter 10 and part of Chapter 11 that deals with muscles. Now, before we get started on the muscle unit, there are three things to point out. The first thing is that the muscle chapter, unlike the other chapters we have done so far, the muscle chapter does go back all the way to the beginning, and it requires knowledge of active and passive transport. So the sodium-potassium pump that we looked at as active transport, things like diffusion, facilitated diffusion, all of that will come back indirectly in this chapter or in this unit. Likewise, the second thing to keep in mind is that we have part of Chapter 11 that will be included in this unit on muscles. It was mentioned on the conversion chart within Eagle Online. I'm just repeating it here just to make sure we're clear. The third thing that we want to be aware of is that I have a video, a short 5-minute or so clip designed to explain how the action part of the muscles work. So the action part of the muscles. So it is important to know the story, but then since you can't explain a story, you have to watch it in a video form. So I have a video available for you to watch for the muscles. Okay. All right. Now we have the first slide here on functions of the skeletal muscle. Body movement. Bones do not move. It's really the muscles that make the bones move. So number one, function of skeletal muscle is body movement. Number two is posture. Posture. Now with posture, I'm going to use three words that come up later. That is posture, tension, and tone. So posture, tension, and tone. That is the second function. The third function is protection and support. The next function says regulating elimination of materials. We can shorten that by saying sphincters, and that's the word that you see on the slide. Sphincters are ringed muscles. You will see that again later. So sphincters are ringed muscles. And as a result, the sphincters will act like a gate, a doorway. So when you swallow, there are actually sphincters along the way that ensure that food and drinks go only in one direction. So sphincters are ringed muscles that open and close and ensure that things move in one direction. You have sphincters in many places, even within blood vessels that you'll learn in A&P 2. Sphincters are even within your blood vessels. All right, the last one there is heat production. So when your muscles are active, that is why you are warming up. So heat production. These are the functions of the skeletal muscle. Okay, next. The next slide talks about characteristics. Now, a lot of people call this characteristics, features, or properties. So on an exam, if someone says characteristics, features, or properties, they're talking about this list. The first two I'm going to take together. The first one says excitability. Now the slide only talks about one of the storylines, but we'll see that there's another story as well. Now one of the mantras that we need to establish starting with this chapter, which is for the muscles, along with the nervous system, which will be our last unit. Muscles and nerves, and notice how I'm saying this, muscles and nerves will operate on what's called electrical and chemical. It happens to be my mantra, which is electrical and chemical. Electrical being the nervous system, and chemical, which is the endocrine system through hormones. So if you learn this well and understand it, it will carry over quite well into A&P 2. So electrical being the nervous system, and the chemical being the hormones by way of the endocrine system. So yes, muscles can be stimulated electrically and chemically. The next one says conductivity. This is the ability for muscles to conduct electricity. You may have heard of the TENS, where people put electrodes on the person's arm, and by turning the electrical current on, it actually makes the muscles contract and stimulate and twitch. You may have seen it on some infomercials that they have now for that too. Okay, well, that's what it is. It is actually an electrical current stimulating the muscles. If you're a big Jason Statham fan, you may have seen all his movies of Crank, Crank 1, Crank 2, Crank 3. These are all showing you how he's using a jumper cable in the movie to jumpstart his heart. Well, heart is a muscle, but guess what? It does conduct electricity. So muscles do conduct electricity, and that's what number 2 is here, conductivity. The next one is contractility. That is the ability for the muscle to shorten. So muscles can shorten by way of contractility. The next one after that is elasticity. Muscles are like rubber bands. They even feel like rubber bands. Okay? So muscles are elastic. They will stretch and they will shorten. So they're elastic. The last one is extensibility. It is the opposite of contractility. Remember, contractility is when it shortens. Extensibility is when it's going to stretch. So these are the characteristics, features, and properties of skeletal muscles. Okay. Do I care for this? Not really. So I don't care for this slide. Okay, this picture is very important. You will find this kind of picture in both your textbook as well as the PowerPoint slides kind of thing. Now, this is the organization that you need to know. They will guaranteed ask this in lab as well as lecture. So we're going to start with the top. Here I have the tendon. As you know, a tendon connects muscle to bone. So tendon will connect muscle to bone. Then we have the entire skeletal muscle. And notice there are different coverings here. So each one of these are connective tissue coverings. So each one is a connective tissue covering. The skeletal muscle, the entire muscle, is covered by what's called epimysium. Okay? So the entire skeletal muscle is covered by a tissue, a covering called epimysium. That's the entire skeletal muscle. Then the skeletal muscle itself has a tube. And notice in this picture here, they're pulling out one of these tubes. And these tubes, this whole black outlined item, is called a fascicle. So in the scheme of things, we have the skeletal muscle containing many, many fascicles. So each skeletal muscle contains many, many fascicles. Then the fascicle itself, so the fascicle itself has a covering. Right? So the fascicle itself has a covering called perimysium. Perimysium. So are you noticing that the skeletal muscle has a covering called epimysium? Then inside of it we have fascicles. Fascicles have a covering called perimysium. Now if I take the fascicle, and this is an entire fascicle here, and I take one of those, I get this rod. This entire thing is a muscle fiber. Some people call it muscle cell. Some people call it muscle fiber. The muscle fibers have an outer covering called endomysium. So are you noticing that is really a tube within a tube within a tube? So we have the entire skeletal muscle, sorry, We have the entire skeletal muscle covered by epimysium. Then inside of a skeletal muscle, we have fascicles covered by perimysium. Fascicles have muscle fibers or muscle cells, and they are covered by endomysium. So it's a tube within a tube, and each of the tubes has a different covering. Okay, next slide. So what I've done so far is I've gotten to this point. Muscle fiber, muscle cell covered by endomysium. What I'm going to do next is talk about this guy that's been pulled out. So we'll talk about that next. This slide is exactly what I've just said, so you can use it if you wish. Okay. We've already talked about tendon, and I already told you that a tendon is something that a muscle is connected to a bone with. So muscles and bones are connected by way of tendon. Don't care about these slides. This is somewhat of an FYI. There are a lot of blood vessels and there are a lot of nerves within a muscle. Okay, next slide. This slide I don't care for because we're going to do it in picture form. Don't care. This one I do. Now, if you look at the top picture, you can see that the author has shown something that we just went over. So that's why I said it's a continuation. So we have our skeletal muscle covered by the epimysium. We have our fascicle covered by perimysium. And we have our muscle fiber covered by endomysium. Now, Nimish, I don't see endomysium here. I am looking at a muscle fiber, this big thing, muscle fiber, but I don't see endomysium here. Here's a reason for that. Just below the endomysium, so just below the endomysium, is something called a sarcolemma. Okay, so something just below that. So just below the endomysium, we have something called sarcolemma. Now, a sarcolemma is nothing more than a cell membrane. If you really listen carefully, you heard me say that muscle fiber is basically a muscle cell. And what do we call the general term for the covering of a cell? We call it a cell membrane or a plasma membrane. So all we're doing now is giving it a special name because it's dealing with a muscle cell. So a muscle cell or a muscle fiber has a covering called sarcolemma, which is just underneath the endomysium. Okay. Alrighty, I'll do it one more time, then we'll move on. So here's what we did so far. We have the entire skeletal muscle covered by epimysium. Then we had a fascicle covered by perimysium. Then we had a muscle fiber or muscle cell covered by endomysium. And then the muscle fiber or muscle cell has another covering underneath called the sarcolemma, which is really the cell membrane. Now, do you see all these holes? All these holes here. These holes lead down into the muscle. Now, they're really called openings for T tubules. So all these holes that you see here and all these holes you see on the side, they are openings for what's called T tubules. So openings for T tubules. Okay, what else I see here is I see a little pillow here. That's a nucleus. So this big thing right here is a nucleus. And then if I look carefully again, further, I see a little pillow looking thing down here, a different color pillow. Those are called endoplasmic reticulum. Now, Nimish doesn't say that here. It says sarcoplasmic reticulum. Well, it's the same thing. It's really an endoplasmic reticulum, but we call it a sarcoplasmic reticulum when it comes to muscles. Now, what's in it? These little pillows that are inside, shaded, the sarcoplasmic reticulum, which from now on I will call it SR. The sarcoplasmic reticulum contains calcium. And yes, calcium is very important for muscles. Okay? So sarcoplasmic reticulum will contain calcium. Now what you're going to notice is I'm actually setting up the chapter with different parts. And then once you know the parts, the other separate video that I have available to you on muscle contraction will show you the physiology of it in terms of how it works. So again, the SR contains calcium. Then, if I go further, I see that I have even more things I can pull out. Well, I've got another rod that I can pull out. And this one's now called myofibrils. So a muscle fiber or a muscle cell has myofibrils. So each one of these in yellow color is a myofibril. Now myofibrils are made out of myofilaments. So myofibrils are made out of myofilaments, and we'll talk a little bit more about that in a little while. So myofibrils made out of myofilaments. The other thing that the author mentions before I go any further, and I already mentioned nucleus, which I showed you up here, but it also says a lot of mitochondria. So muscles have a lot of mitochondria. And if you recall from the earlier chapters, a cell has mitochondria for ATP production. So your muscles need a lot of ATP energy. So that's why you see a lot of mitochondria within the muscles. Okay, now let's move down. So I've already mentioned that myofibrils have what's called myofilaments within it. So on the far right at the bottom, you actually see the myofilaments, these little yellow and purple. So yes, we have a lot of tube within a tube within a tube kind of setup. Now on this myofibril, shown down here as an example, I have these little zip line looking things, little Z's. If you look carefully, they look like little Z's, like a zipper. Well, from one zipper to another zipper, and they often call it Z disc to Z disc, that is one sarcomere. So that's a unit. So a sarcomere is a unit from one Z disc to another Z disc. So that's one unit. They call it one sarcomere, from Z disc to Z disc. Now, a sarcomere goes by another definition. That is, it is the basic structural functional unit of a muscle. So that's what a sarcomere is. It's the basic structural functional unit of a muscle. So that's a sarcomere. Let me go to the left here, and you can see the T-tubule story, which we'll see a little while later. Then we see our sarcoplasmic reticulum, which I already told you stores calcium. So that's so far a summary of what we've done. And now we see our myofilaments that I'm going to go into in the next slide. So again, we have a tube within a tube within a tube structure. Okay, now what is this slide? Let me back up for a second. So right here, we're going to talk about that sarcolemma up here and these myofilaments. Now, what the author is showing with this slide is here's my sarcolemma, that outer covering, that sarcolemma. And notice it has a positive and a negative. Well, at rest, when the cell or the muscle in this example is doing nothing, we learned, and we should have learned before, that the outer part of a cell is typically positive. The inside of a cell is typically negative. That's what is shown here. The outside being positive, the inside being negative while at rest. Then we see our channels. Remember I said at the very beginning of this slide, or this video, that I mentioned that we have the sodium-potassium pump kind of thing coming back? All right, well, that's what this is. Okay, so the sodium-potassium pump will play a role. The channels will play a role. The ion movements will play a role. Okay, all old stuff coming back here applied. Now what this is telling us is that while the muscle is at rest, the outside charge is positive, the inside is negative, and we're going to have something happening with the sodium and the potassium. Here's that T-tubule that you saw earlier that goes deep into the muscle. Now what I'm giving you now is a forethought. What we're going to have is we're going to have an electrical current going down this sarcolemma. Now a electrical current going down is called an action potential. So that's basically an electrical current. Now, Nimish, is that an important term for both now and in the nervous system chapters? You bet. An action potential is an electrical current. It's not the same kind of electrical current you have in the outlet of your house, but it is an electrical current created by the exchange of sodium and potassium, and that's going to create an electrical current, and we call it an action potential. And because here we're talking about muscles, it's going to be called a muscle action potential. When we get to the nervous system chapter, we're going to call it a nerve action potential. So Nimish, if I do well and understand the muscle chapter, will it help me with the nervous system chapter? You bet, because the storyline carries over. So a muscle action potential is very much related to a nerve action potential. They are both currents. A muscle action potential is a current on a muscle. Nerve action potential is a action potential on a nerve. Okay, next slide. This slide is a repeat of what I've said. You can look at it at your leisure. Okay, here's the sarcoplasmic reticulum. You can look at this at your leisure. I will be mentioning these two terms, but they're not important yet. So I'll mention these two terms. They're not important yet. And we said that sarcoplasmic reticulum is the storage, the storehouse for calcium. And these two terms will come up at the very end, much later. Okay, next. This slide is something I'm going to show by picture form, so it's not important for you. Same as for all of this. All of this will be shown in picture form so we can move forward. Again, every time I skip a picture or a slide like this, it is there for you to read, but it's also something that I'll show in picture form. So here's the picture form of that. So we have our story again, the muscle fiber, muscle cell with a rod coming out called myofibril, and the myofibril has myofilaments. Now there are two main kinds of myofilaments. We have the thick filament. The thick filament is made out of these little golf club looking things. Many authors, including your textbook as well, will call myofilament here containing the thick filament to be like a golf club. So these are your myosin heads, part of the thick filament, and they look like golf clubs. Now these thick filaments containing myosin, well, that's where ATP will react. That's why you see it up here. Okay, so one more time. Myofilaments are made out of thick filament. The thick filament has these myosin heads. Now, what do you think myosin's made out of? Something from a previous chapter. We learned that proteins play multiple roles. And I called it a verb in the past. So if you watch any of my previous videos, I said that they are a verb, an action, so proteins. So myosin is really a protein. Okay? So that's what I've done so far, describing the thick filament containing these myosin heads, which look like golf clubs. And that is where ATP will eventually bind to. Then the other kind of filament that I have as part of myofilament is the thin filament. Now the thin filament is made out of three things. The first thing I see is tropomyosin. I call it the tropomyosin belt. And as you can see, it looks like a green colored belt here. So tropomyosin belt. the green little belt. Then I see troponin, and to me it looks like a snowman. So troponin, this little troponin piece. And then most of these things here are the red balls made out of actin. So the red balls are made out of actin. All of these things that make up the thin filament are also made out of protein. So actin, troponin, and tropomyosin are all made out of proteins. Remember, proteins do things. So the thin filament is basically protein as well. Now again, we have these three parts I've called out. Now let's look at what they do. Here I see the tropomyosin belt. And if you look carefully, see how the actin has these black dots? You can see these black dots on them. See how at the present moment this tropomyosin is covering the black dots? Right? Well, those black dots are called myosin binding sites. So what's going to happen is this golf club looking thing, these myosin heads, with the presence of ATP, they're going to be able to stick to these black dot areas, but not at the moment. Why? Because at the moment, this tropomyosin belt is blocking this binding site. Well, when will this belt be moved out of the way? Well, do you see troponin here in our calcium story? When calcium sticks to troponin, so when calcium binds to troponin, the belt will move out of the way and expose this myosin binding site. And then myosin can stick to it. Now, Nemish, are you sort of describing the steps that go in that video? Yeah. But Nemish, are there steps I can write down? Well, don't be bothering to write down those steps now. Why? Because I've already provided the steps under the modules section of Eagle Online. There's a handout. And that handout has all of the steps. And if you're the sharp student that has already printed the handout out, you'll notice that some of the stuff I'm describing to you are already on the steps handout. For example, I mentioned that calcium will eventually bind to troponin. And when calcium does stick to troponin, the tropomyosin belt will move out of the way and expose the myosin's binding site. And then the myosin heads, with the presence of ATP, can then stick to this myosin binding site. So the handout with the steps along with the video will be very, very helpful for this chapter. Okay, next slide. Okay, here's a repeat of what I've said. I said that a sarcomere is a Z disc to Z disc. And I also told you it's the basic unit of a muscle, right? It's the basic structural functional unit of a muscle. Next slide. Now, I have I bands, A bands. This slide is nice, but there is a picture coming up. You will want to know the picture, both for lab class and for the lecture class, because they will want you to know these bands. And in the slide that I'm on now won't help, because you really want to have a visual of it. Okay, these are nice words, but a visual is better. So here's a picture of the visual of it, showing you the various bands. Okay, that is going to help you for lab and lecture. Okay, so this picture is for that purpose, the different bands. There's your sarcomere again from Z disc to Z disc, and now you can see the language. Z disc to Z disc is one sarcomere. Then we have our I band, A band, H zone, and then the M line. And there's a better picture coming up. And here's the better picture. Okay. So I'm now going to go through this picture. So we have our Z disc to Z disc. That's one sarcomere. We have our thin filament shown in this position. That's the actin, the troponin, the tropomyosin. That's all the thin filament. Here's my thick filament with the myosin golf club looking thing, the myosin protein. Here's the M line, which is the center line. And then we have this green one. This green one in this author's book, remember I'm using a different book, this author calls it connectin. Some books call it connectin. Some books call it elastin. Be aware of both because you don't know who is asking the question. So connectin or elastin. Now what does this connectin or elastin do? Its job is to help the muscle come back to the same starting position. So when you actually contract a muscle, and then when the muscle relaxes, you don't want it to over-relax. You want it to come back to the same starting position. So connectin or elastin's job is to make sure I don't overstretch or I understretch. I always come back to the same starting position at rest. And that's what connectin and elastin will do. Okay. All right, next slide. I don't care for this one. All right, so connectin was already mentioned. Okay. Now the other one I want you to know for disease purposes is this one down here called dystrophin. This is the one that is damaged when someone has muscular dystrophy. So if some of you are old enough to remember the Jerry Lewis telethons, I actually came to a TV network with a jar of pennies and walked across the stage. So I got to see myself on TV walking across the stage with a jar of pennies to give to muscular dystrophy. I was a big fan of Jerry Lewis back then. So muscular dystrophy has a problem with this protein called dystrophin. Okay, so be aware of that. They do ask that on department exams and things like that. So dystrophin. So you have connectin and we have dystrophin. Okay, this one I don't care too much about now because it does come up later, but I'll quickly mention a one or two liner on this. Okay, myoglobin is a type of structure that holds extra oxygen. So yes, you have hemoglobin in your blood that holds oxygen, right? Because you got oxygen going around in your blood and it's stuck to hemoglobin. Well, your muscles need a lot more than that. So your muscles have what's called myoglobin. Now here's a rule that's going to help you for a future slide. More myoglobin makes the muscle more red. Okay, so just remember that as a tip for later. More myoglobin makes your muscle more red. So if your muscle is a whitish color, that means it has less myoglobin. Does that make sense? So more myoglobin will make the muscle more redder. Less myoglobin will make it more whiter. Then down here we have glycogen. Now you learned this in one of my earlier chapters. And we learned that glycogen is stored in the muscle. So it is an energy storage in the muscles. Glycogen. Very, very important this time around. Then the last thing we see on this slide is creatine phosphate. Now someone will say, especially these bodybuilders I get once in a while in my class, will say, is that related to that creatine that bodybuilders take? Yes, it is. Okay, it is related to that. Creatine phosphate is a shortcut energy form, but it does not provide a lot of energy. It just provides enough energy for a short burst to do something. That's it. So creatine phosphate is the cheapest energy source. And if you look at this slide carefully from bottom up, we have creatine phosphate, which is the cheapest and shortest energy. Glycogen, which is anaerobic, right? Anaerobic, which is the second level. And then we have myoglobin, which is aerobic, which provides for the long-term energy. So creatine phosphate is a quick and dirty energy that does not last long. Glycogen is next in terms of being a little bit better, but it is anaerobic. And then the best energy form is, of course, aerobic. And we'll see that later. Okay, now comes this slide. Motor unit. This definition is very important. A motor unit is a motor neuron in all the muscle fibers it controls. Okay, so a motor unit is a motor neuron in all the muscle fibers it controls. In this example, I see two. I see a purple one and a reddish one. So the motor neuron that goes with purple will only excite these two that are purple. Then the ones that are in red are going to be stimulated by the motor neuron that's red, and it'll stimulate 1, 2, 3, and 4 because those are the 4 red ones. Now the way it works is that we tend to use the cheapest or the least motor units first, or weakest. Some people even call it weakest. So if I want to pick up something light, like say a stapler, then I'm going to use the purple ones in this example because I'm using the least amount of motor units. So lifting something light will use fewer motor units, like in this case the purple ones. If I start lifting something heavier, so let's say I have one book that I'm holding, it's a lighter book, so I'll use the purple motor unit there, the 2 purple ones. If I add an extra book, so I have now 2 books in my hand, since it's heavier, I'm going to have to use more motor units. I might use one more of the red ones. If I add another book, so now I have 3 books, I might have to add yet another of the red. And if I have another book added to my hand and starting to get really heavy, I'm going to have to use even more motor units. And of course, as you know, that if there's too much weight in my hand, I might not have enough motor units left over, and whatever I've done, I've maxed out the muscle, and now I can't lift that item anymore. Okay. Well, that's what I'm getting at. We use the fewer motor units first, and then we start adding more and more motor units. Now, there's a term for that. It's called recruitment. Recruitment is the adding of more and more motor units. So we use the fewer motor units and the weaker ones first. So the fewer and the weaker ones first. And then we start adding more and more motor units as the case in the case of the books. Right? So more weight will require more motor units, and that's going to be called recruitment. Now my next comment is important. Your fingers, for example. You have a lot of dexterity in your fingers. Well, your fingers have many, many, many small motor units. That's why it's nice and flexible. Okay? That's why magicians can do all their little sleight of hand. So many, many, many small motor units will be for dexterity. Some people call it fine control, and that's what I'm leading up to. So for fine control will be many, many, many small motor units. Okay, now the other example, your thigh. Okay, you don't really do much with your thigh muscles except lift your leg or something like that, right? So your thigh is going to have large motor units. Okay? So they're going to have fewer but large motor units, and that is for gross control. So if it's just gross control, then it's going to be few but large motor units. So let me say it one more time for clarity, and then I'll move on. So your fingers, which have a lot of fine control, will have many, many, many small units. That's for fine control. Gross control, like your thigh, is going to have small but larger. When I say small, yeah, it'll be small or fewer, rather, actually fewer. So let's say that fewer but large motor units. So your leg would have fewer but large motor units. Okay, next slide. Now what I just said to you is now here on this slide. So if you want precise fine control, you're going to have many small motor units. Okay, that's for precise control. If you want gross control, then it's going to be large but few. So it'll be fewer but large motor units. Okay, and that's what this is. This is just a nice comment at the bottom. Just be aware that these motor units are not shown like in this previous slide where all the purple ones are next to each other and the red ones are next to each other. That's not really the case. All they want to let you know is it's really clustered all the way around. So it's dispersed evenly around the muscle. Not a big deal. Okay. Alrighty, next slide. Okay, this slide's important. Okay, now your muscles are stimulated by nerves, and it's shown up here. Here is a motor neuron, and these are the muscle fibers. So motor neuron, muscle fibers. Well, just like a nerve connecting to a muscle or one street connecting to another street, there is some kind of space in between. Well, that's what this is. So if you look carefully at this picture, I have a motor neuron coming down, and then there's a little space in between. They're not touching. There's a little space in between, and then you have your muscle down here. Now, Nemish, is that similar to your fingers, your fingertips, hovering over a table? That's exactly what it looks like. So your fingertips would be the motor neuron. So your fingertips will be the motor neuron. And then they're hovering just above the table. The table would be the muscle. Okay. Now, do you see this arrow here? This arrow is a nerve action potential. That's what that arrow is. Here they call it a nerve signal, same thing. So remember how I said that a nerve action potential is basically a current going down a nerve? Well, that's what this orange arrow is. It's a nerve action potential. You will find it on my handout for the muscle steps. Then we have a dead end. Well, just like many neighborhoods have a cul-de-sac, that's the dead end street. Well, think of it the same way. And that's why I gave you your fingertip example a moment ago. When your fingertips are hovering over the table, those fingertips are the synaptic knob. Now, I'll keep calling it synaptic end bulb because that's the other name that some people are still using. So a synaptic knob or a synaptic end bulb would be your fingertips that are hovering over the table. Then we have our muscle down here. That space I mentioned in the middle, that's the hovering of your fingertips over the table, has a little space in between. That's called the synaptic cleft. So the synaptic cleft is the space between your fingertips and the table when your fingers are hovering over the table. There's our endomysium. Remember I said that an endomysium is covering a muscle fiber or muscle cell. And then I said just underneath it. So just underneath the endomysium is our sarcolemma. So see what I'm getting at with this particular slide that we're on. It's a summary of all those structural things we saw earlier. So we've got our muscle cell or muscle fiber. It has a covering called endomysium, and just below that is the sarcolemma. All right. Now, what about these dots? These little marbles. Well, those dots are going to be a chemical, a neurotransmitter. Okay, a neurotransmitter. So chemicals are going to be released across this synaptic cleft. So chemicals are going to be released across that synaptic cleft. Okay, then the next thing we have is this area here. See how there's a bracket? Well, that bracket is called the motor end plate. Well, that motor end plate contains receptors. And those receptors are for the neurotransmitter that is released above. So from these little white dots, chemicals called neurotransmitters will be released, and they will go across the synaptic cleft, and then they will go to the receptors that are found on the motor end plate. Now, Nimish, that's nice for me to imagine, but is there a slide showing a picture for that? Yes, it's coming up. Okay, so again, these white dots contain neurotransmitters. They are chemicals that will go across, and they will bind to receptors that are found on the motor end plate. So notice I'm using words like receptors, channels, all the old stuff. Okay, here's an enlarged view of the same thing. So up here at the top is my synaptic knob or synaptic end bulb, just like the fingertips that are hovering over the table. This is the space I mentioned before between the fingertips and the table, called the synaptic cleft. There is my sarcolemma. So this is the sarcolemma. Again, a sarcolemma is nothing more than a cell membrane. There's my motor end plate. So this is my motor end plate containing the receptors, and these little grayish things are receptors. And here's how the storyline unfolds for this. And again, all of these things are part of the steps to a muscle contraction, the handout. Up here at the top, we have a nerve action potential coming down this motor neuron. Now, in the handout on the steps to a muscle contraction, you'll find that, guess what happens? An electrical current has the ability to move things. So an electrical current can cause a chemical to come out of something. Likewise, when I move chemicals, like moving sodium and potassium and whatnot, moving ions can also create an electrical current. So that's one of the things we need to master as we go forward, that moving chemicals creates electricity, and electricity can move chemicals. So in this storyline, what we see here is that electrical current, nerve action potential, is going to come down this motor neuron. And in doing so, do you see these little calcium particles here? These calcium pieces are going to go out and be pumped out by using a calcium pump. So these calcium molecules will be pumped out. Doing so creates a gradient. And now, by moving it out intentionally with active transport, pumping the calcium out is going to make calcium want to go back in, and that's going to create an electrical current that's going to cause this vesicle. And remember what a vesicle is. It's anything that moves things around, like a transport container. And this vesicle contains a neurotransmitter called ACH. The full form of ACH is acetylcholine. It's a key main neurotransmitter. Well, this changeover here, the storyline, this electrical current that's created, is going to cause ACH to be released from the synaptic end bulb. And this ACH is going to flood into the synaptic cleft, that space between the fingertip and the table. So now ACH is flooding this area. And ACH is going to stick to these receptors. You can see that on the next slide and a little bit closer on this slide, that ACH is going to stick to these receptors. When ACH sticks to the receptors, it's going to cause it to open up like a channel. So as you can see, it's opening up here as a channel. What's going in? Sodium is going in. What is coming out? Potassium is coming out. So sodium goes in, potassium comes out. That is your sodium-potassium pump in essence. So sodium is going in, potassium is going out. Now, if you have caught on to what I've said, then that's a very important detail. Are you telling me that as sodium goes in and potassium comes out, and you just said that moving chemicals can create an electrical current? Is that what you're telling me? That's exactly what I'm saying. So as sodium comes in and potassium leaves, we're creating a muscle action potential on the sarcolemma. So we're creating a muscle action potential on the sarcolemma. So here's what we got so far on this slide. We had a nerve action potential coming down the motor neuron, and then some chemicals are moving as a result, and in this case it would be like ACH. And when the ACH goes across and moves across, it creates sodium and potassium exchange, and that sodium potassium exchange is going to create a muscle action potential on the sarcolemma. That is exactly what I'm saying. Now, Nimish, did you say that sometime earlier? Let's go back a few slides. If you recall, when we were on this slide, I said the same thing, that we're eventually going to create a muscle action potential. And yes, I said the same thing just now. We are creating a muscle action potential on the sarcolemma. So what do you think is going to go down this T-tubule that I did not mention before, but now I am clearly saying, is that what's going to go down that T-tubule? Muscle action potential. An electrical current is going to go down this T-tubule. Okay, moving back to where we were. Okay, so this is the slide we were on. Okay, this slide is basically the words of the picture we just talked about. This slide says the same thing. So these are all words that go with that picture. Same thing here. Now look on this one. Look what it says. Initially we're at rest. So initially we're at rest, called resting membrane potential. At rest. But because we opened up the sodium potassium channels, we have now created a muscle action potential on the sarcolemma. Okay, so if I continue, okay, so there it is. So in this picture, what do you see in the orange arrow? The orange arrow says action potential propagation. That's a fancy way of saying that a muscle action potential is moving down the sarcolemma. And then, as I showed you earlier, and I mentioned a moment ago too, what goes down the T-tubule? Muscle action potential goes down the T-tubule. That is correct. So muscle action potential goes down the T-tubule. Now, Nimish, how come it has these little calcium red dots? Well, if you recall, I told you that the sarcoplasmic reticulum is a storage for calcium. So, Nimish, how does that story unfold? Well, it goes this way. It's basically saying that the muscle action potential that went down the T-tubule is now stimulating the SR to release calcium. That's right. So that's what we've done. All this calcium is now flooding into that muscle space. It's flooding into that muscle space. Now, what do we normally, in general, what do we normally call that open space in a cell? The cytoplasm, right? That's what we normally call it. Well, in the muscle, we just have a different name for it. It's called sarcoplasm. So all of this calcium is going to be sort of flooded into the sarcoplasm, the open space in the muscle. And that calcium is going to come from the SR. So the SR is now releasing all this calcium into the sarcoplasm, into that open space. Okay, so it's going to flood this open space. Okay, again, words that go with what we just said. Here's another picture of the same storyline. So the author sort of goes back a little bit and explains the same thing. So we have our nerve action potential coming down the motor neuron, going to the synaptic bulb or the synaptic knob, causing the calcium to be pumped out and then brought back in, which allows the vesicle to release ACH. So the vesicle is releasing the neurotransmitter ACH, and then it goes up to the receptors, and this is just another view of that previous story. Okay. Now we have myasthenia gravis. That's just a medical disease. And when it comes to these clinical questions, some of the clinical questions are important. So like this one is important. Now the problem with myasthenia gravis is that it's an autoimmune disease. So it's an autoimmune disease. That means the person's own immune system, so the person's own immune system is destroying the receptors. So if I go back a moment, remember how we have these ACH receptors? Okay. And let's count how many I have on this example. 1, 2, 3, 4, 5, 6. Well, what this says in the disease of myasthenia gravis is that the person's immune system is actually destroying these receptors. Now here's why it's important. If I do not have enough of these receptors opened, so if enough of these receptors don't open, then I might not get enough sodium to come in. And remember, it's the sodium that comes in and the potassium that leaves. That's what creates a muscle action potential. Now, one of the laws, and we're going to have 3 laws, and the 3 laws are true for a muscle chapter, and the same 3 laws are true for the nervous system chapter. And one of the laws is all or none. And the all or none rule says that once a muscle action potential forms, it either forms or it doesn't. It's like a light switch. It's either on or it's off. There's no in between. Okay? Same way here. If I have enough sodium going in and I trigger a muscle action potential that goes down the sarcolemma, it's either there or it isn't. This is where the disease comes in. What if I don't have enough of these receptors stimulated because they are damaged? So I have 1, 2, 3, 4, 5, 6, and let's say 2 of them are damaged. Then that means that muscle fiber, the one we're talking about in this example, that one muscle fiber won't fire. That means you're going to have a muscle weakness, and that's what myasthenia gravis is all about. It usually starts off in the face and the head with the eyelids and things like that. Then it works its way down through to the neck, and then the shoulder and the arms and legs, and eventually it gets to the lungs and the person dies because they can't breathe anymore. Well, that's what this is about. So myasthenia gravis is a problem with the receptors going bye-bye because of an autoimmune disease, and that causes muscle weakness. Okay, and that's what this slide is all about, the one we were on originally. So they get a weakness. It starts with the facial muscles typically first. So this is a slide to be aware of. You do need to know how this disease comes about, meaning what's the information I just shared with you. Okay, next slide. Here's a repeat again. The author likes to have a lot of these slides. Now notice the title here, excitation, contraction, coupling. In my video on the muscle contraction, you're going to see that it has multiple steps. And one of the steps is titled excitation, contraction. So the first one's of course going to be excitation, and the second one's going to be excitation, contraction, coupling. Somehow we have to get excitation connected to those myofibrils, the myosin and the actin and all that. That's what the upcoming slides will talk about. Okay, next slide. This slide is now something you should understand. We have a muscle action potential going down the T-tubule. The muscle action potential that goes down the T-tubule will cause calcium to come out of the SR, and calcium will flood into the sarcoplasm. That is what we have so far in our story. So calcium has come out of SR and it's now flooding the sarcoplasm, which is this open space right here. Next slide. I don't care. It says the same thing. I really don't care for this because it's beyond what we're going to worry about at the moment. So words like depolarization, we're not going to worry about right now. I will tell you this. Let's do this much. Depolarization goes with sodium influx. That much we can say before we go any further instead of writing all of this. So depolarization goes with sodium influx. When sodium is going in, we call that depolarization. So here's how that should be broken down. The physical act of sodium going in is depolarization. But the word depolarization is the electrical term. So physical is influx. So sodium influx is physical. The electrical word for that is depolarization. Okay, what about the other one that you see on the slide? Repolarization. That's when potassium leaves. So when potassium leaves, we call that outflux. So potassium leaving is outflux, but the electrical word for that is repolarization. And what you've learned so far from me is that it takes a D followed by a re to make an action potential. And that's why I said I don't care for the full slide, but the information I shared with you is enough. It's enough to get you going for the A&P1's nervous system chapters. Next slide. Don't care. Okay, this slide I do care. Now on this slide, I see a flat line all the way. That flat line means we are at rest. So that flat line means we are at rest. No sodium is going in, no potassium is leaving, it's at rest. Then we reach a peak. So some of that sodium is going to come in. Now if you recall, I said that one of the rules we had for action potential was all or none, which means once I reach action potential, it's either on or it's off. The second rule, both for a nervous system and for the muscle, is threshold. Threshold. That means I have to reach a certain threshold before an action potential is created. So we have the first law so far. Again, it's true for muscles and for the nervous system as well. The first law was all or none. So once I reach threshold, then I'm going to have an action potential or I'm not. That's all or none. The other one was threshold. That means that muscles and nerves must reach a threshold. That means a certain amount of sodium's got to come in before it fires. And when I say fires, it means creating an action potential. So in this picture here, I am at rest, at rest, at rest, and then all of a sudden, I have just enough sodium going in to fire it fully because I reach threshold. And once I reach threshold, I will have depolarization, which means sodium will rush into the cell. And then once I get to a peak, sodium will stop and potassium will start to leave the cell. Remember, potassium is outflux. It will leave the cell. And notice it overshoots it a little bit. Now that overshooting is important. It's called hyperpolarization. Remember, it's true for muscles and nerves, so you'll see it again in the nervous system chapter. It's called hyperpolarization because I went below my original starting point. So it's hyperpolarization. You do not want to leave it there because if you leave it there, that muscle, or in the nervous system chapter, that nerve will no longer fire, and that's a bad thing. So what you want to do is you want to have the sodium-potassium pump come in and reset it. So the resetting is done by the sodium-potassium pump, and it's going to bring it back to that resting membrane potential of -90. Now that last trailing bit that I mentioned is true for muscles as well as the nervous system chapter. So hyperpolarization is when you go below the original peak, That's bad to leave it there. So the sodium-potassium pump will kick in and bring you back to resting membrane potential. That part is important. Okay, I don't care. We saw this already in the picture form. We already said that calcium will leave the sarcoplasmic reticulum, and we have already said so far that calcium will get into the sarcoplasm. And that's what we've said so far in our overall story. Okay, now comes the continuation of the story. So far we've got up to that point. So now, what about the calcium? Well, calcium, as you can see, is now sticking to troponin. And if you recall in a previous picture, I've mentioned that once calcium sticks to troponin, just like this, the tropomyosin belt will move out of the way. And when it moves out of the way, what is now exposed? The myosin binding sites are now exposed. That means that the myosin heads, the ones that look like a golf club, can now stick to these binding sites. Now, in order for it to stick to it, I have to have ATP. That's not mentioned yet. It's coming up. All right, next slide. So now we see the language of the ATP. Again, I don't like to copy too much of the wording, so I have a picture for you. Now, the way it works is going to be like a mime. You know how a mime, when he wants to pull a rope, he grabs hold of the rope and then pretends to pull it. Then he lets go with one hand and attaches it to the rope again and pulls like a mime does to pretend like he's pulling on a rope and pulling something that's attached to the other end. Well, the exact same thing happens here. So first we have this situation where calcium is stuck to troponin. The tropomyosin belt has moved out of the way and exposed the myosin binding site. Then notice in the presence of ATP, so you have to have ATP, look what happened. See how it is in this starting position shown by the silhouette? And then when ATP is present, it's cranked. Well, that's what happens. The ATP allows the myosin to crank, attach, and if you look at the bottom picture here, pull. So it's crank, and then the book calls it reset. I call it crank. So crank, attach, and pull. So it takes an ATP to crank, attach, and pull. That's one ATP. So it takes one ATP to crank, attach, and pull. Then, once it cranks, attaches, and pulls, what are we really doing? As you can see in this middle picture here, we're really pulling the thin filament on one end and the thin filament on the other end closer and closer to the center. And if you pull both thin filaments on both sides closer and closer, what happens overall? Overall, the distance between this Z-disc and this Z-disc shortens. That's basically a sarcomere shortening. That's how a muscle contracts. Then I have a fresh, and if you look at my handout on the steps to muscle contraction, you'll see that it takes a fresh, a brand new ATP to release. Okay, so it takes one ATP to crank, attach, and pull, but it takes a fresh, brand new ATP to release. And if I do this whole cycle over and over, where I crank, attach, pull, let go, crank, attach, pull, let go, that's called power strokes. If I go back a slide, you'll see the word power stroke. It's also mentioned at the bottom one down here too, power stroke. So it takes ATP to crank, attach, and pull, and a fresh ATP to let go. So what I'm really doing in a muscle contraction is pulling on the thin filament and bringing the Z-disc on one side and the Z-disc on the other side closer to the center, which was called the M line. And by shortening the distance between that Z and that Z, I've shortened the muscle. And that's what a muscle contraction is. Okay, next slide. And don't forget the word cross bridge, that's the attachment. So a cross bridge is really the attachment. Okay, forms a cross bridge. And remember, it takes ATP to do that. Okay, next slide. These are the words that went with that picture we just looked at. And this picture shows you what's happening. So here's a relaxed muscle where we have a pretty big distance between that Z and that Z. And then when I contract my muscle, like this guy is doing flexing his biceps, when I contract, I see that the distance between the Z and the Z has shortened, and that I've brought the thin filament closer and closer to the center. And notice that they're now overlapped. They're now overlapped. Now, one major test question that people ask is, did I actually shorten the thin filament? And the answer is no. What they're asking is, did I actually shorten the thin filament from here to there? No. What I really did is simply overlapped it. So I'm not shortening the thin filament, but I am shortening the distance between the Z disc and the Z disc. That is a tricky nuance that they're very likely to ask. So you're not shortening the filament, but you are shortening the distance between the Z disc and the Z disc. Okay, now we have these two diseases. What they're going to do on a test is they're going to ask you, do you understand how these two diseases unfold at a certain location? So what is that location called? Now, I intentionally didn't mention it, and that's why I'm scanning back on the slides, is to show you that we had a name for it. It's called neuromuscular junction, and that is that space that we talked about. It is a connection between the nervous system and the muscles down here at the bottom. So nervous system's up front or at the top, and the muscles are down here. That's called a neuromuscular junction. Now, why am I highly stressing this? If you understand how a neuromuscular junction works for this chapter, you'll understand how a neuro junction works for the nervous system chapter, because all you're going to do there is, instead of having a muscle down here, it's just going to be another nerve down here. Does it change the overall storyline any? Not really. So that's why if you understand a neuromuscular junction, then it's very easy. All you do is change a few terms, but it's the same concept as a neuro junction. So this is called a neuromuscular junction. Okay, going back to where we were. So these are the two diseases that operate at the neuromuscular junction. And that's true for a neuro junction as well. First one is tetanus. Now here's a word that people get confused by. Spastic paralysis. That's when the muscle stays stiff and tight. So don't think that paralysis always means limp. It doesn't. Okay. You have spastic paralysis. It gets stiff. So tetanus. Now some of you know tetanus as lockjaw. That's what some people, the old terms, used to use. Lockjaw, where your jaw is now stiff. Well, that's because of this problem. It blocks the release of inhibitory neurotransmitters. So just as ACH excites a muscle, there are also neurotransmitters designed to stop a muscle or to relax a muscle. Well, in this case, you don't have those neurotransmitters to block the muscle. The muscle stays on all the time. And that means that the muscle stays stimulated and the muscle doesn't get a chance to relax. And that's what happens when you have like a rusty nail. And it doesn't really have to be a rusty nail. It's the dirt that's in the rusty nail that causes the problem. It's not the rust, it's the dust, the dirt. And the dirt's got a bacteria in it that does it. Okay, so it blocks it. So this is now the inability to relax. Then we have botulism. Now this is what the cosmetic surgeons are now putting into those little frowny lines that you have on your forehead or on your face with the crow's feet. Botulism is caused by another type of bacteria, and this one causes relaxation, paralysis. Okay. Here, notice it says it prevents the release of ACH. So if no ACH at all goes across the synaptic cleft, that space between the muscle and the nerve, if no ACH goes across, then the muscle never gets stimulated. So it would be like this storyline for what we've learned. So if I go back to my classic picture, if I never have ACH in this space, then this muscle never gets stimulated. And that would cause a relaxed muscle. And that's what your frowny lines on your face, the little crow's feet and things like that, that's what that is. And let me get to my picture. There it is. All right, so that's what that is. So it prevents release of ACH at the synaptic knobs. Now, if you have your muscles never stimulate, like your respiratory muscles, your breathing muscles, then of course you're going to die because you can't breathe. So these are examples of clinical views that they often ask about. Next slide. All right, here is how the muscle relaxes. So on last few steps, I think it's step 16, 15, 16 on my handout. So on my handout on muscle relaxation and contraction, you're going to find these are the things that have to happen. So do go through this slide. And see step by step how the muscle will relax. Now, one of the things I want to highlight is this guy, hydrolysis of ACH by something called acetylcholinesterase. A lot of people call it ACHE as in Edward. Don't confuse ACH with ACHE. Now let me explain how ACHE works. Again, going back to my classic picture. Once a muscle action potential forms, so once I have a muscle action potential moving down my sarcolemma, I want all of this to stop. How do I stop this? Because the muscle action potential is going to continue its story. That's going to cause the muscle to contract. That's all good. Okay, but we don't want this ACH to stay in this synaptic cleft. As long as ACH stays in this synaptic cleft, this muscle down here will make muscle action potential forevermore. And you don't want your muscles contracting forevermore. So what nature says is that once I have a muscle action potential moving down on this sarcolemma, I need to stop all this action that's going on upstream. And one of the things I have to do is destroy this ACH, these little diamonds here. And I do it using the chemical that I'm talking about now called ACHE, which is acetylcholinesterase. It's an enzyme designed to destroy the ACH. And by destroying this ACH here, I no longer make additional MAP unless I start with the brand new storyline with a nerve action potential. So I have to have fresh, brand new nerve action potential whenever I want to stimulate a muscle. You don't want this ACH to stay here forever. Otherwise, one nerve action potential will cause the muscle to contract forevermore, and you don't want that. Okay, so that's one. All right, so I was going back to my slide, and that's this long list of all the things that happen. One of the other things I need to talk about is returning calcium back to the SR. So one of the things I need to talk about is returning calcium back to the SR. Now, in one of my slides that I started with, that I said don't worry about knowing at the moment, is this slide. These two items are the pumps that take calcium back into SR. So calsequestrin and calmodulin. These are the two that are key. So these two are key for returning calcium back to the SR. Okay? And it says it right here. Has pumps that import calcium back into the sarcoplasmic reticulum. And these are the two pumps. Okay, so be aware of that. Those are the two pumps. As you can see, I go back and forth a lot on the slides to join the excuse me, to join the concepts together. Okay. All right, so this is the slide we're on. So again, make sure you know the steps to how a muscle relaxes. It is mentioned on the handout as well. Next slide. Okay, now the author switches gears and talks about the energy source for a muscle. So these are the same three energy sources that I mentioned on a previous slide. The first one was creatine phosphate, which I said was quick, short, provides very, very little energy, which provides very little short bursts of contraction. So it's not going to make a big deal in terms of energy. It's enough energy to lift maybe one more weight one more time. And that's why when these bodybuilders take the creatine monohydrate, it will help, but it won't make you into Arnold Schwarzenegger. All it's doing is providing a little extra energy to lift that weight one more rep out of the maybe 10 reps that you do when you weight lift. Okay? So it's a very little amount of energy. The second one, the next one is short term with glycogen or glycolysis. Now I used the word anaerobic when I described it earlier. So glycolysis is anaerobic. If you learn anything in general biology, anaerobic produces very little ATP, which is only 2 ATPs overall. So very little ATP, and it's anaerobic, without oxygen. And you know that glycogen is stored in the muscles. That's all old stuff coming back. Then long term. This is your ATP by way of aerobic. This is the one that's going to produce a lot of ATP. Something on the order of 1 glucose making something like 32 ATPs. Some people say 34, some people say 36, some people say 38. Don't get bogged down on that. There is a general biology reason for it, which is beyond the scope of this class. But be aware it's in the 30s. So 1 glucose, when 1 glucose is fully burned for energy, it produces about 36 to 38 ATPs, but it's done aerobically. That means with oxygen. And earlier we saw a slide that says myoglobin provides that extra oxygen. And the more myoglobin, the more red the muscle will be. The less myoglobin, the more white it will be. Okay, don't care for this slide now. Okay, this is the stuff I just went over, so be aware of this slide, but I'm not going to regurgitate it again and again. So be aware of this slide. Again, glycolysis, glycogen only producing 2 ATPs. Next slide. Again, it does not have oxygen, so that's why glycolysis was anaerobic. There's glycogen there too. Okay, next is this slide. Here it says 30 ATPs. Remember I said something on the order of 36 to 38 ATPs overall. It is aerobic, it requires oxygen, and it's going to use a lot of energy for this, or produce a lot of energy for this. And it is aerobic. It does use myoglobin. Okay, I don't care. This is just to let you know the same thing I said. If you want short sprint, short burst of energy like lifting a weight one more time, that would be the creatine phosphate route. If you want a little bit more energy, it would be the glycolysis route. And if you want a lot of energy, like if you're doing the decathlon or the Ironman is going to use aerobic. Okay, I don't care. Here's a nice picture if you want to use it. Oxygen debt. Okay, this is a nice list that they ask about. Oxygen debt has to do with once you have exercised and you got to now pay back. Like, just like when you borrow from a bank, you got to pay back. Well, that's what this is. So when you are exercising and then you're finished your exercise routine, the heavy running around, weightlifting, whatever for the day, you took your shower, you're now sitting on the sofa, relax and calm down. This is what's going on. So after you exercise, you're actually taking in extra oxygen than what you would normally breathe in at rest. So you're actually more efficient at getting oxygen into your body after the exercise. Why do you need all that extra oxygen? Well, that's where this list comes in. This is the reason why you need all that extra oxygen. One, you got to put it back onto the hemoglobin and onto the myoglobin. You got to replenish your glycogen. You got to replenish your ATP reserves. And you have to convert the lactic acid back to glucose. So when you do exercise, whether you're exercising aerobically or anaerobically, mostly it's anaerobic, like 99% is anaerobic, but one way or the other, when you are exercising, you're going to produce some amount of lactic acid. Because you always start off with anaerobic and then work your way to aerobic. So you're always going to produce some lactic acid. Now this lactic acid is what's causing the muscle ache after you exercise. So if you're not breathing properly, while you're on the treadmill, for example, and you're holding your breath instead of breathing properly, you're going to produce a lot of lactic acid. It takes the liver about 48 hours to get rid of the lactic acid. Is that important for Nemesh's test or anyone else's test? Yes. So it takes about 48 hours for the liver to take care of that lactic acid. That is important. Okay, next slide. Okay, so I took care of that. Okay, now comes the type of muscle fibers. Okay, so the type of muscle fibers. I don't really care for this slide. I just stopped here to give you that title. So we have 3 types of muscle fibers. I will only be stressing 2 of them on a test. Okay, what about the department exam or anybody else? Same thing. Most people will not bring the one in the middle because it confuses people on the test. So it's going to be either one or the other. So it's either going to be slow or fast. So yes, we have 3. So be aware of it that we have 3. But we're going to focus only on the 2 types. So we have slow fibers and we have fast fibers. Now, any given muscle, so let's say your thigh muscle. Your thigh muscle is going to have either more slow or more fast. We all have both, but some people have more slow, some people have more fast. Now, who decides whether I have more slow or more fast? One of the things is genetics. So if I mentioned Kenya, Ethiopia, okay, what are they good for? What are they good at? They're good at long distance running, right? Jamaica, Ethiopia, Kenya, they have a lot of good runners, right? They often are first, second, or third in a marathon race because genetically they are blessed with having more certain type of fibers. So who decides whether you have more slow fibers or fast fibers? Genetics. Genetics will play a role with that. Okay, now here are the 2 names down there. We have fast twitch fibers and we have slow twitch fibers. Now the fast twitch and slow twitch, here's how it works. We'll do the fast twitch first. Fast twitch are more powerful. And I'm just reading that line at the bottom at the moment. Fast twitch are more powerful. However, because they are quick and fast, they're going to run out of ATP quicker, sooner. So a kid that has a bank account, but he always likes to buy things and buy expensive things over and over, he's going to run out sooner, right? Same way. So a fast twitch fiber, it's more powerful, it's quicker, it's faster, but it is going to run out of steam sooner. It's going to run out of ATP quicker. And because of that, it's going to be anaerobic. Okay, anaerobic. So fast twitch fibers tend to be anaerobic. Now let's see how sharp you are. Because I said fast twitch fibers are anaerobic, then what color do you think they're going to be? They're going to be white. So fast twitch fibers tend to be white. Why? Because they do not have much myoglobin. They have very little myoglobin. That's why they're white. Because they have very little myoglobin, that means they're going to produce, because they have less oxygen, right? Less myoglobin means less oxygen, which makes it anaerobic, which makes it have less ATP, which makes it run out of energy sooner. You can use the same logic for the slow twitch. The slow twitch are going to be less powerful. They're going to be red. That means that they are aerobic. They're going to have more myoglobin, hence the reason for them being red. And that means they're going to be aerobic because they have a lot of myoglobin, which means more oxygen, which means that they are going to have a lot of ATP. All right, and therefore more energy. And what kind of advantage will a slow twitch fiber provide? Endurance, long distance. So if you are blessed with slow twitch fibers, like the Kenya, the Jamaica, and the Ethiopia, they have more slow twitch. That means they have more red muscles. They're going to have more endurance, and they're going to be able to do the marathon races. The fast twitch fibers are for the sprinters and the bodybuilders. So fast twitch tends to be bodybuilders and sprinters, whereas slow twitch tends to be the marathon runners. Okay, next slide. Now what I just shared with you is now on this slide. So oxidative, that's going to be fatigue resistant. All right, so oxidative, fatigue resistant. Aerobic, having a lot of myoglobin. And since it has a lot of myoglobin, it's going to be red, a lot more mitochondria. And so we're really describing the slow fibers here in this paragraph at the top. Then at the bottom, we're describing glycolytic because it's anaerobic, less mitochondria, less myoglobin, so they are white. Okay, that's how it works. Okay, next slide. This is what I mean by the three types of muscle fibers. We have the slow, we have the fast, and then we have the middle one. The middle one is hardly ever asked about, but just be aware that it exists. So you need to know the slow, you need to know the fast. Slow makes sense because it's oxidative. That means it has to do with oxygen, aerobic. And there's the word aerobic. Fast glycolytic means glycogen. So therefore, it's going to be anaerobic, and there's your word down there, anaerobic. Fast is going to be white because of less myoglobin. Slow at the top is going to be more red because of myoglobin. So make sure you're aware of the number 1 and number 3 for the test. Okay, I really don't care too much about this slide. I already told you that the proportion, whether you have more red or more white, or whether you have more slow or more fast, is going to be determined based on genetics. And there it says it right there at the bottom. I've also given you this information in my comment a moment ago. Long distance marathon runners tend to have more slow. Sprinters and bodybuilders tend to have more fast. It's determined primarily by genetics, in some way by training, but primarily by genetics. It is scattered throughout. Next slide. Okay. Now we go into the next change of topic, which has to do with tension. Now, if you recall, I mentioned the three words, posture, tension, and tone at the very beginning. There are two things that play a role in this posture, tension, and tone. I'm mentioning it now. I'll mention it again when it comes to the right slide. But posture, tension, and tone, one of the reasons for posture, tension, tone will come from the nervous system. So your nervous system will play a role. The second reason for this posture, tension, and tone will be something I talked about called connectin, which I mentioned some books call it as elastin. Remember I said that connectin or elastin is what allows the muscle to come back to its original state. Now this slide right here has a lot to do with my steps and along with the words that go with the video. So here I have my flat line. Then right here it says stimulus. Then there is a flat line that continues relatively. And then I have it spike up. Well, this latent period is, excuse me, your latent period there is when we're electrically stimulating the muscle, but the muscle hasn't contracted yet. So what do we call that? We call that excitation, right? So in my handout, we had excitation, and then we have something called excitation, contraction, coupling. Those two really belong to this latent period. Now on the handout that has the steps, I have a word there that says lag time or latent period. I believe it's step number 12 on the handout. So if you have your handout present in front of you, you probably can see it with me. So steps 1 all the way to step number 12. So step 1 to step 12, what are we doing? We're only exciting, and we're exciting and contracting, or setting up the contraction rather. And that's called excitation, contraction, coupling. And that is what's going on in the latent period. And you'll see that step 12, that says steps 1 through 12, right? That's what step 12 says. That steps 1 through 12 were really the latent period or lag time. And latent period and lag time is really the excitation and the excitation dash contraction coupling that you find in the video. Then we have our contraction state, and then we have our relaxation state. Okay, next slide. Okay, so this is what that is. I don't really care for this slide. Okay, this one is important, and we did talk about it. We called it recruitment. I mentioned it at the top of this video. Motor units were the motor neurons and all the muscle fibers it stimulates. And as I add more and more motor units, we called it recruitment. And that's what this is. So basically, my tension of my muscle, the contractionness of my muscle gets stronger. Notice it does, of course, max out. Because you can't lift a car with your one hand, can you? Arnold might, but we can't. Because we're using all of our muscle fibers, all of our motor units, and there's nothing left over, and therefore that's the reason we can't lift the car. Okay, next slide. I don't care for this one. Okay, this one I do. And this one does show up in both the muscle chapters and later on in the nervous system chapter. This one is called TREP. Now notice how it works. So let's describe this one. I don't care for the language on the left. I do want you to know the description I'm providing. So here I have a starting point. It is contracting and it is fully relaxing. Contracting, a little bit stronger in the contraction, fully relaxing. So in TREP, we do allow it to come back to full rest. That's what we see here. We have a stronger contraction, but we allow it to fully rest. Why? Because the frequency is not too fast and not too slow. So the frequency is going to determine how strong these contractions are. And in this case, the frequency is just right, enough to do what? Allow it to come back to rest. Now if I go to my next slide, you'll see that it is different now. In this slide, and the language to describe it is here, but I don't like it because it's too technical and too confusing. So I like the picture here. It's very similar. The first part is called wave summation. Why is it important for knowing it now? Because there's something similar to that in the nervous system chapter as well. So notice what happened. I had a certain frequency that in this case I did what? While it's contracting, everything's good. But then while it's relaxing, I zap it again. I don't let it rest. So while it's starting to relax, that's when I zap it. So notice, while it's starting to relax, I zapped it. So it's again contracting, of course stronger. Again, while it's starting to relax, I zap it. So it contracts. Again, I don't let it relax. Zap it so it gets stronger again. So notice my frequency is starting to increase. And as I do so, I don't really let it relax. So I'm zapping it. Notice each contraction is now stronger. So this one is getting stronger, this one is stronger, and each time I do that, I do it just when it's relaxing and I don't let it relax so the next one gets stronger. Now if my frequency is so high that I eventually can go into tetany. Here the frequency is so high that I'm not letting it rest at all. So no rest at all. Now what eventually happens is that I run into fatigue. So if I zap it so much that I run into fatigue. Now fatigue by definition is when I run out of ATP. I'm contracting so strongly that I don't have enough ATP. Remember how it works? In order for me to do this, I have to have what? 1 ATP to crank the myosin head, to attach to the myosin binding site, and then to pull, and then I need a fresh ATP to relax. Notice I'm not getting to relax at all. There isn't any ATP left, so eventually I cannot stimulate this muscle at all. It's going to be stiff, 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 and then it's going to relax. Now, two things to be aware of. This chapter primarily is focusing on skeletal muscle. So skeletal muscles will have tetany. Skeletal muscles will have fatigue. That's very, very important. So skeletal muscles do have tetany. Skeletal muscles do have fatigue. That is not true for cardiac muscle. Very, very, very important. So cardiac muscle does not have tetany. Cardiac muscle does not have fatigue. You will see that again when it comes to A&P2 with the heart chapter, right? Cardiac muscle goes with the heart. So no tetany, no fatigue for cardiac muscle. Okay, moving on. Okay. I don't really care for this slide on tone. I already mentioned posture, tension, tone. Don't care for this slide. Okay, I do like this slide. So this slide is worthy. You do need to know the difference between isometric and isotonic. But many books do have a picture. So I'm going to go to that picture in a moment. So isometric contraction, isotonic contraction. And under isotonic, I have two stages. So under isotonic, I have two stages, concentric and eccentric. Let's go to the picture. So this is the picture that goes with it. Let's first do the isometric on the left here. Isometric is this guy right here holding the baby in this position and this position only. Here, he is contracting his muscle, and guess what? It's not going anywhere. It's not stretching. It's not shortening. He is holding it steady. This is like you leaning against the wall with your arm straight. If you have a straight arm and you're just leaning against a wall, that is isometric. If you are doing Pilates and you're doing the plank, right, where you're steady, you're not going up and down like a push-up, you're just holding a plank position, that is isometric. Okay, so when you're just having a straight arm, leaning against a wall, or you're doing a plank in Pilates, you're not moving. So isometric is when the muscle length does not change. So the muscle length will not change. Okay, that means the tension is less, because all you're doing is just holding your body up in a plank position or leaning against the wall. You're not doing anything really for your muscle growth. So if you do a plank all day, all it will do with your muscles is provide it tone, tension, and posture. So planks are good for your posture, but it's not going to make you into Arnold Schwarzenegger, right? It's not going to build bigger muscles, for example, okay? Because you're not changing the length of your muscles in isometric. No net movement. It says it at the bottom. No net movement. So that's isometric. Isometric will not make you bigger. It'll provide posture, tension, tone, but that's it. Now let's go over to isotonic. Isotonic is when the muscle length does change. So when you're lifting weights, where you're lifting weights up and then down, up and then down. So if you're doing a bicep curl and you're lifting the weight up to your shoulder and then away from your shoulder, like he's doing with the baby here in this picture. Now we have two stages of isotonic where the muscle length changes. The first stage is muscle shortening, and that's concentric. So when the muscle length shortens, that's concentric. When the muscle length is lengthening, that's eccentric. Eccentric. Okay, you will need to know that. Now when it comes to this baby, for example, it is much tougher for him to bring the baby towards his shoulder or towards his head, because he's actually lifting the load of the baby. So concentric is a little harder because you're actually muscle length is shortening and it's much heavier because you're actually lifting the load. The easier process is eccentric, where you're lowering the baby down towards your foot. So eccentric is when the muscle lengthens, and that's a little easier. Now, for the longest time in PE, I could never climb that rope and ring the bell at the top of the gym. I couldn't do that. So I had problems with my upper body strength lifting because of my back and shoulders and all that. So I got me my little iron gym thing that hangs on the doorway. And at first I had trouble lifting it up. I used a little stool, used my legs a little bit to lift myself up. More of my legs got the workout than my arms did. Then eventually I got the little elastic bands to help me with the pull-up. Then I got to a point where I can just jump up and hold myself. And what I learned is doing the eccentric part, which is the lowering part, is the muscle lengthening part. That was easier. So I could jump up to the top and slowly lower myself down with control. And that actually helped me do the other part, which was the concentric. So easier is eccentric, concentric's a little harder. And now, of course, I can do it. I can even do L sit-ups and L pull-ups on the pull-up bar, which I couldn't do in my earlier years as a kid. As a senior citizen, I now can do it. That's a little shame, but that's how it works. Alrighty. Do I care for this slide? Not really. I don't care for this one. Okay, don't care for that language. I do care for this picture. So what you can't happen or have happen is you don't want to stay contracted. You don't want to stay too far stretched. You want to be like Goldilocks, right in the middle. So who is going to allow us to always come back to the right rest? You don't want to come back to this as a rest. You don't want that to be as a rest. What do you want to do? You want to come back to the middle as rest. And what two things did I say allow for that? The nervous system and the connectin, or if you're using a different textbook, they call it elastin in many books. Okay, that's what it is. That's all you got to get out of that. Okay, fatigue, I've already talked about it, is when you run out of ATP. So I don't really care for this slide, other than knowing you ran out of ATP. Okay, don't care. Don't care. Okay, I do care for this word up here, hypertrophy. Now, hypertrophy is an increase in the size of the muscle. Okay, increase in the size of the muscle. Now, the reason why the size of the muscle increases is because the size of your myofibrils, that sarcomere, all of that has increased as well. Okay, so hypertrophy is an increase in the size of the muscle. Now, listen to this example. This is very important. Arnold Schwarzenegger and I have the same number of muscle fibers in our biceps. So my biceps, his biceps, for Arnold Schwarzenegger, we have the same number of muscle fibers. The difference is the size of my muscle fiber and the size of his muscle fiber. He's got more stuff in it. He's got more mitochondria. He's got more myofibril stuff. But the number of muscle cells, or the number of muscle fibers, stays the same. Now let me give you an example that'll help you with this. When you go to Taco Bell and you order a bean burrito, okay, so you go to Taco Bell and order a bean burrito. If you go when they're really, really busy, they really don't fill that bean burrito well. So you have less filling inside that single bean burrito. If you go when they're not busy, then they fill it up with a lot of extra cheese, a lot of extra beans, a lot of extra onions and whatever. They put it and they fill it up. So I've got now 2 bean burritos in front of me, one when they were busy and one when they were not. And as you can see, it's 1 and 1. So that means that the number of muscle cells, or the number of muscle fibers, is 1 and 1. But the stuff inside of it is going to be different. The busy time is going to have less filling. That means it's going to have less of these motor units and all of that. That means it has less of that stuff. That means it's going to be able to lift less. Whereas hypertrophy is the increase in the size. All right. Now comes atrophy. Atrophy is both. Not only loss of number, but also loss of size. You want to make sure you clarify that here. So atrophy is both loss in number of muscle fibers, so loss of muscle fibers, as well as in the size of the muscle fiber. So you're losing both. So if you're in a cast a long time, or if you're in space for a long time, like the Kelly brothers that came back from space, one of them is now a senator. Well, if you come back from space after a year or two, yes, your muscles are going to waste away. And you're going to have loss of muscle fibers as well as the size of the fiber as well. So atrophy. Okay, so this slide is a value. Okay, now aging. When you get to the AARP age, yes, I'm a member of the AARP. I promised that when I was a high schooler. Once I can join AARP, I would. When you get to a certain age, you're going to lose muscle fibers. That means you're going to have less endurance. That's why the old geezers can't run as long or as far. That's why if you ever watched a marathon with the senior citizen group, they always take like almost the entire day to finish because they're just so slow. Right? Their reaction time is slow. Their endurance is less. They got less muscle fibers, less diameter, less everything. Less oxygen carrying capacity, so it's bad. Okay? So be aware of the top half of this when you age. And that's true, by the way, of every chapter in A&P 1 and in A&P 2. All the chapters end with what happens as you age. Okay, I don't care too much about the anabolic steroids thing. Okay, now I'm coming to this slide, cardiac muscle tissue. instead of me going through all of this, there is a chart found in every textbook at the end of the textbook. So every textbook at the end has this story about the different types of muscles. Now, in a previous chapter, we talked about the 3 types of muscles. So remember in the muscle chapter, in the tissue chapter rather, in the tissue chapter we talked about the 3 types of tissues. You will need to know that chart again. So that's what this is. So make sure you know the skeletal muscle, the cardiac muscle, and the smooth muscle. Okay. So if you remember, cardiac muscles have these characteristics that were mentioned in the tissue chapter. So the tissue chapter talked about these. So the intercalated discs, the desmosomes, and the gap junctions. They are found in cardiac muscle. Cardiac muscles were striated, that type of thing. Cardiac muscles are autorhythmic. So again, features from the muscle, the tissue chapter: cardiac muscle, skeletal muscle, smooth muscle. So make sure you know the features. So you have striation for cardiac muscles. You've got intercalated discs, desmosomes, gap junctions. You will need to know these for A&P 2. So when you get to the heart chapter in A&P 2, your instructor will ask you about these 3 again: the intercalated discs, the desmosomes, and the gap junctions. Autorhythmic. So the heart muscles are autorhythmic. The heart does beat and does have a rhythm of its own when it's both inside the body as well as outside the body. Okay, so cardiac muscle important. I don't care about the picture. Okay, smooth muscles. Again, go back to the tissue chapters chart, and every textbook's got a chart at the back. Make sure you know the features that go with smooth muscles as well. Now, smooth muscles are found anywhere you have fluids moving. Now, when I say fluids, it doesn't have to be a liquid. It could be a gas. So your bronchial passageways, your lung passageways, they have smooth muscles. That's why someone that has asthma has to take asthma medicines to calm down these smooth muscles, to help them breathe. Okay, so smooth muscles are anywhere a fluid is moving. So like in your lungs, you have smooth muscles for your airways. Your blood vessels have smooth muscles because it's moving blood, right? Because it's moving a liquid, blood, it's going to have smooth muscles in your blood. Okay, your digestive tract. So when your food is going down into your poop, well, the smooth muscles are there to move food and stuff down your digestive tract. Your PP, your urinary tract. Your urinary tract also has smooth muscles as well. Okay, so these are the examples I basically gave you. I just called them out, but they're here on a slide as well. So blood vessels having smooth muscles, airway in your lungs having smooth muscles, your intestines having it, your urinary system having it, and your reproductive system having smooth muscles when it comes to delivering a baby, for example, in the uterus. So these are all smooth muscles. Here's an example in picture form that many books use. So smooth muscles, smooth muscles. Here's a picture version. Okay, now on a test, there is a one-liner that you find on many, many charts. And this one-liner is what I'm now mentioning. Smooth muscle is unique. It does not use the calsequestrin. Remember earlier I said calsequestrin is being used? Smooth muscles use calmodulin. So let me get to that slide so I can show it to you. Okay, so I'm going to keep going, and it's not on the slide yet. It's coming. And there it is. Calmodulin. So not calsequestrin, but calmodulin is for smooth muscles. It's a one-liner. It shows up on a test sometimes. There it is in bold, calmodulin, and that's unique to smooth muscles. So remember I said calmodulin and sequestrin are what takes the calcium back to SR. Well, that was a generic statement. So calmodulin and sequestrin are generally what takes calcium back into SR. But for smooth muscles, it's going to be calmodulin. And that's why I'm giving you that clarification right now. So see what it says. Unlike skeletal muscle, smooth muscles use calmodulin. Skeletal muscles would use calsequestrin. So skeletal muscles, calsequestrin. Smooth muscles, calmodulin. Okay. Don't care. Don't care. Don't care. And no, I don't care. Do I? No, I don't. I don't care. Don't really care. Don't care. Don't care. Don't really care. Don't care. Don't care. Don't care. Don't care. And that takes care of that.